Hi, thank you for coming. I am here to talk to you about salmon lice larvae. So for my PhD, we've been working on trying to quantify these little larval parasites within the fjord system. So we're measuring their distribution within the water column and their dispersal from and towards salmon farms. So for anyone who's not super familiar with them, they're copepod ectoparasites. They've been an issue now for the industry for a few decades, but they've existed for salmon long before that. They have a direct life cycle, so they're living just on this salmonid fishes, but they have these 3 early life stages where they are drifting as plankton in the fjord systems. So we're really interested in quantifying these individuals, figuring out where they are, what they're doing, what kind of behavior is driving their dispersal, so we can know a little bit more about the risks associated with different farming techniques. So we do our bulk plankton sampling so that we can get enough of them to actually quantify via passive plankton sampling, so this one in the middle here, where we leave a net out in the fjord for days at a time. We rely on the currents to push water through it and capture the larvae for us. We've also used a few different types of water pumps and active pumping methods, and I'm going to talk about that as well today. So when we do this, we filter, tens to hundreds of thousands of liters of water, and we end up with this sort of mass of plankton, 250 mils in this case, upwards of a liter. And this is a big soup of plankton, and it's way too much to process under a microscope. So we're homogenizing it. We're putting matrix beads in it, shaking it up, breaking all the cells apart, and removing or releasing all the DNA. And then we're subsampling it. So we're taking a small portion of that, we're extracting the DNA, we're then amplifying it, but only for our target species. And then we're able to estimate the number of DNA copies that we find within that sample. And then based on that, we're able to come up with an estimate of the number of organisms there. So we do that based on the first chapter of my PhD, where we calibrated the number of DNA copies per individual. We did it for 2 species, but today we'll just talk about Salmonis. And we found that there were about 2.5 million copies per individual at any density. And so that's the number we're going with. For our fieldwork that I'm going to talk more in depth about now, we chose a site that's about 4 or 5 hours west of here in Storfjorden. So it's quite an active farming region. In the last few years, it's had more lice problems. And NTNU Ålesund conveniently has a research license for this farm at Gudmundset. So we used this facility in a beautiful part of Norway with great accessibility, labs nearby, and we deployed our equipment. We decided to do 6 different sites, looking at different distances from the farm. We also deployed our nets at 4 different depths because we were quite interested in seeing where the lice were in the water column. And we did that because the currents are different depending on how deep you go. We can model their dispersal from the farm, but if we don't actually know what depth they're in, it's not going to be very reliable. So this farm has predominant surface currents to the west, but as soon as you get down to 10, 15 meters, it's a lot more chaotic. So with that in mind, we did our samplings. We did 1 day, so 24 hours. We did a really wide opening size, and we did this on 4 different occasions. Yeah, here's the results. There's a lot going on here. It's very chaotic. There's a lot of colors. But the gist of it is that we captured lice at all depths. So those 4 depths that you can't really read over there behind the flowers, the blue is 2 meters, red is 6 meters, green is 12 meters, and yellow is 18 meters. So we're seeing lice at all depths and at all distances from the farms. So really, we're having quite good effectiveness in capturing the larvae, and we're also able to tell a little bit about how they're actually being distributed when we look into it in a more statistical manner. So looking at some zero inflation models to counter the fact that we had about 50% zeros in our samples, we saw that there was an effect of distance, there was an effect of depth, and there was also an effect of the direction, so the current there. We also saw some statistically significant effects when it came to the conditional model, so the number of individuals rather than just the presence and absence. So we had a lot of lice at the sites closest to the farm. We had more lice at the sites to the west of the farm, and we had more occurrences of lice actually deeper than 2 meters, rather than what we thought would be the case, which was more lice in the upper column. So when we look at the currents, it gets a bit more complicated. We have, in this first deployment, really strong western currents. We see a lot of lice on the west side of the farm, but then when we get to these other cases, we've got sort of a mix when we're at the surface versus deep down. This one, we've even got some that are sort of pushing the lice at the surface offshore, away from the farm. And it's a lot harder to read how those are directly affecting the lice that we're seeing within our samples. So we really need to do some more collections of very fine-scale dynamics of the currents around these farms. So we also did pumping. We did these deployments of equipment away from the farm, but we also did some directly at the farm. So we did nets, or we did pumps inside and outside of the net pens, both with and without lice skirts. And then we did pumping during and after the production cycle, trying to see what was going on during the fallow period. So we got a lot of lice, upwards of 600 in some of these samples. So granted, all of these ones that are purple, these are inside the pen with lice skirts at 2 meters. So the lice larvae aren't really being allowed to disperse as they would naturally. So if we remove those and we have a look at the points with a slightly different distribution here, we can see that there's a lot at all the depths, double, triple digits. In 3 hours of sampling, these were typically like 30 cubic meter samples. So we're seeing the most at 9 meters, but also at 12 or 2 and 18, and really not a big difference between day and night. Now, if we want to look at these in more detail, we want to look at the difference between nauplii and copepodites. And in order to do that, we're using some FlowCam and machine vision technology that's being developed at NTNU Ålesund by our collaborators. And they have a really effective method of determining the larval stage that we're seeing in our samples using this technology. Now, the ddPCR is really good at species-specific detection. It's not so great at stage-specific. We're working on that, but it's not quite there yet. The FlowCam is really good at stage-specific detection, but there's quite a few species of lice out there, and they're pretty hard to discern when you have the resolution that we have to deal with in FlowCams. So together, we can test and verify these methods, and we see they actually agree really well. We have a little bit of overestimation maybe by the AI or underestimation by the ddPCR when you get into this really, really high values. But overall, we have a really strong positive correlation between the two methods, and we know that based on the processing of samples with traditional microscopy, both of these methods blow it out of the water. 100 times or 1,000 times more volume. It's just far more effective, and we really want to highlight that these methods should be being used for site-specific verification or site planning purposes, they're really effective at determining the number of larvae that are present in water and figuring out the densities and the risk. But now, if we look more closely at both of these together, we can see that these values on the right, they're much, much lower. These are all still positive values, despite being right here near the 0 axis. But that's because this is after the last fish was slaughtered, and it's more than 2 weeks. So none of these individuals are from lice that were on the farm. These are all individuals that are sort of background lice levels for the fjord system that we're in. So they're either coming from other farms in the region, or they're coming from wild fish. And we really wanted to look at these and the differences between the nauplii and the copepodites in these samples. So these are all the occurrences that we had of copepods. It's not that many, but it's at least enough that we can see that there's quite a few lice at depth and not a strong temporal change. So these are the ones that was confirmed by both methods. The ones on top were actually not processed by the AI, sadly, but we're going to exclude that one anyway because there wasn't a lot of DNA. It's probably just a fragment. So going forward, we can see that there's not a strong difference between day and night, really none that's detectable. It's a low number of individuals, but we're not seeing any pattern there. When it comes to depth, we're definitely finding more in 2 meters, like 60 to 75%, depending on which ones you count as validated. But that still leaves 25 to 40% that are present in deeper waters. So these are copepods that in most cases we're assuming are right at the surface. So we really need to be accounting for the fact that these individuals could be at 9 meters, they could be at 18 meters, they could be much deeper where we haven't even measured. So some things to take away. Lice larval densities definitely decline as we go further from the farm. That's expected. The predominant currents are driving large-scale dispersal, which again, we expect, but there's definitely some fine-scale dynamics at play, and we need to be taking those into account. The lice larvae as a whole, so that's probably mostly nauplii, are more abundant at depth. And this also needs to be accounted for in our dispersal models. And the copepodites, which they're more abundant near the surface, it's proven in lab studies, it's proven in mesocosm studies, and we're finding it as well, but they're also at depth. So they're not just at the surface. And we need to keep that in mind when we're trying to plan for sites that we're using avoidance measures. So things to consider are that although depth avoidance, submerged cages are super effective. We can reduce the number of lice tenfold, and the number of delousings can decrease significantly as well. So they're definitely a fantastic technology, but we need to know that there's still risk. It's not just like you put the fish down there and the lice won't get to them. So we need to do some local site forecasting in determining the risks that the lice will get down there. And the technologies that we've developed and others are developing as well are going to be really imperative in doing this. So just to sort of some takeaways from this conference for me have been that the innovation in this sector is incredible. Walking through these exhibitions, it's phenomenal what people are doing. But the original innovator is evolution. And lice larvae are being pressured by the technologies we're using, and they are rapidly developing resistance. And the same is going to be true for depth-based installations. So it's really important that we're choosing installations where we're less likely to have larvae disperse there so that we can prevent a higher number of individuals developing resistance in that way. So with that, just thank you to the many, many collaborators who helped with the project at NTNU Trondheim, Ålesund, and Nina. And thanks to you all for listening. Please give me questions if you have any.